Swayam Prabha. Digital India. Educated India.
In this lecture, I will be talking about emerging aspects in microbial geotechnology and ground improvement. This area of geotechnology where you are trying to improve the ground using the microbes has been something that people have been pursuing in the research, and I felt that some of the advanced topics also need to be understood in this area of ground improvement, because in this ground improvement area we have a lot of traditional techniques that we discussed in the course so far, and also the emerging techniques like this, and many emerging applications as well, which are supposed to be very sustainable. And in this talk I would be talking about the applications. As I just mentioned, you have this microbial geotechnology is something that has come and that is emerging in the recent times, and I would be talking about two applications, namely bioclogging and biocementation. Bioclogging is nothing but the you are trying to produce pore filling materials. For example, in sands the permeability of the material is going to be high, but if you by introduction of this bacteria, if you are able to have some clogging, then the permeability of the material comes down. As a result, the seepage losses are also going to be less, and it is going to be more stable. So, this is an interesting application, and normally we use clay in the case of if you want to reduce the permeability of sand, and but then both this clay, when clay is not available, or it's a more a sustainable material to use bioclogging. The second thing similarly is that you are trying to introduce cementation. Normally we use cement in the case of some materials to introduce cementation in the form of bonds, but if you are able to get the same cementation effect using some sort of bacteria, then it is going to be very nice. So, you are trying to introduce these concepts of bioclogging and biocementation, which is something that they are like we can say that they are more sustainable technologies, because bacteria are always available in nature, and at the same time you are able to have a good advantage of these materials. There is some the reference for this work, actually I must draw from the papers of Ivanov and Chu, who have, particularly Chu has been working in the area of ground movement a lot in this area, and he has contributed significantly, and this is a paper from one of the journals of environmental science and biotechnology, and the thought for some of these ideas have been articulated by Professor Mitchell, who is an authority in the area of soil behavior, and Santamarna, who is also a professor in Georgia Tech. These two professors have a lot of vision, and they envisage use of biomaterials or bioclogging or biocementation or use of biotechnology in the area of geotechnology. This is what it is, and in this connection we need to identify suitable microorganisms. They are of different types. One is facultative anaerobic bacteria, facultative anaerobic bacteria, microaerophilic bacteria, anaerobic fermenting bacteria, anaerobic respiring bacteria, obligate aerobic bacteria. There could be different types. In fact, it is said that the billions of bacteria that are present, the count is so much that it's an unexplored area in the area of biotechnology. What are all these means? Maybe from engineering point of view, I would like to just make some simple differentiation. It is said that aerobic and anaerobic bacteria can be identified by growing them in a liquid culture. For example, if you take a tube and then leave them, and the possibility is that if the bacteria needs oxygen or they survive more in oxygen, they come up. So, that is what I meant by aerobic bacteria. but then if they go down away from the water, then you can say that it is anaerobic bacteria. So, then there could be some bacteria which is called facultative. They can be everywhere in general, and they are they are generally occurring. Then the other one was that that's what it means, that they do occur in general and particularly in anaerobic conditions. And then microaerophilic bacteria, what it means is that they are more towards attracted towards this is particular one, this first one corresponds to obligate aerobes, obligate anaerobes, and then the facultative anaerobic organisms could be like this. Microaerophiles, they are close to, they want the presence of air. Sometimes another one you have air tolerant one. So, one can understand that the suitable microorganisms that one can have in soils are that like facultative anaerobic bacteria, microaerophilic bacteria, anaerobic fermenting bacteria, anaerobic respiring bacteria, obligate aerobic bacteria. So, there are some of these listing is given, and one should have a look at what the size of the bacteria is. People are aware that these are all very, very small size living matter, and they have in one can say that their range is in terms of the mm's to micrometers to even nanometers. People are even questioning, do you have nanobacteria? So, definitely you have a bacteria of very small sizes in the range of millimeters to micrometers, and if you look at the scale, in terms of millimeters, micrometers and nanometers, and some of the atoms and polymers and molecules do come here, but when you are trying to use with soil, you should understand the grain size distribution and the particle size in soils. So, definitely the clay minerals have certain like say for example 75 less than 75 microns, we can call them as clays, and you have silt and sand materials. So, definitely there is some sort of relationship, one can definitely the size of the bacteria is somewhat compatible with the sizes that you have, and one can really look at this as a means of understanding the interaction between clay minerals and the bacteria. So, if you look at this size, the size beyond this particularly, so this area corresponds to silts and sands, and here what it means is that unhindered microbial motion and easy nutrient transport. So, it is going to be quite effective, but if you want, the size is going to be very fine. Microorganisms cannot pass pore throats, but may be entrapped during formation. So, if you want to use it properly, so you need to really distinguish these two, that the size has a big role in understanding the use of bacteria in soils. What are the factors that affect the application of microorganisms? The first and foremost thing is you must be able to screen and identify suitable microorganisms. There are too many, and you must be able to identify the right ones for the right purpose. Optimization of microbial activity in situ. When you are trying to apply in the field, there are so many variables, and you need to put the right quantity, so that you have that effect obtained. Then biosafety of the application is something that is also very important, like safety is an issue, particularly when you are trying to deal with biomaterials, and it should not lead to complicated problems like viruses and other things. So, cost effectiveness is something that is very important, that you should consider for large scale applications. Maybe for laboratory studies and exploratory studies it is all right, but then when you are looking at large scale applications, one should see that the whatever biomaterial or the technique that you have got needs to be cost effective. So, then you should also see the stability of soil properties after biomodification. So, what happens to the soil properties after you modify them with biomaterials, bacteria? So, the factors affecting the applications of microorganisms are, you need to really understand that you have a couple of issues here, and see that they are satisfied to get the maximum effect of these materials. I would just mention about bioclogging. The bioclogging is something here is to reduce porosity and hydraulic conductivity of the soil and porous rocks due to microbial activity of products. So, it is where do you use it? Its use is to use as a sealing material for leaking construction pits, landfills or dikes. Wherever there is a leak, definitely the porosity is more and you would like to reduce the leakage. So, you put this bioclogging. Then there are some cases where use as a grouting material for soil erosion control, mitigating liquefaction and also enclosing of bioremediation zone. So, what it means is that you can use these materials as a for bioclogging. So, similar to use as a grouting material. Grout material can be used for mitigating liquefaction and also for enclosing of bioremediation zone. So, for example, you have a bioremediation zone, you need to be contained in a particular area, you don't want you want to isolate the area from the next available areas. So, you can use bioclogging. Examples of bioclogging could include microbial production of water insoluble polysaccharides in situ, and of course, it involves high cost, production of bacterial exopolymers in situ, and this is another thing. So, if you just look at detailedly in this table, of course, it may not be visible, but one can read from read the original papers. In this table, there are different items mentioned. One is physiological group of the microorganisms, what type of groups they belong to. Say for example, algae, aerobic and facultative anaerobic bacteria, then oligotropic bacteria, nitrifying bacteria, sulfate reducing bacteria, ammonifying bacteria, iron reducing bacteria. There are many types of bacteria, and I must confess that one should have a good knowledge of the biotechnology or microbiology here, and it is always better to interact with a microbiologist who understands your requirements and also the help in the experimentation at a laboratory scale to see what effect they have in the actual response of the soil. When you have so many types of bacteria, then the mechanism, what is that mechanism that one can have? The mechanisms of bioclogging can be a few, like in some cases it could be formation of impermeable layer of biomass, like a formation of impermeable layer of biomass is formed. Then there is the production of slime in the soil, and the production of slime in soil, the production of slime, precipitated matter, production of undissolved sulfates of metals, and formation of undissolved carbonates of metals in soil due to increase of pH and release of carbon dioxide, production of iron or ferrous solution and precipitation of undissolved ferrous and ferric salts and hydroxides in soil. So, there are some various mechanisms that one should really look at it in terms of the analysis, and then look at what is going to happen and what could be the likely mechanisms when you add certain bacteria. Then once you add certain bacteria, the thing is there are essential conditions that are required for effectiveness of this system. So, you take a particular bacteria, say for example algae, it needs light penetration, presence of nutrients. Algae cannot grow without the presence of light and nutrients. The common example is algae growth in the lakes. Then aerobic and facultative anaerobic bacteria, presence of oxygen and medium with a ratio of carbon nitrogen more than 20. And then in some other places it could be low oxygen, low concentrations in oxygen. So, nitrifying bacteria, presence of ammonium and oxygen in soil. So, some of these things have been mentioned here, and one needs to understand that yes, these conditions have to exist to have maximum benefit. Then when you have these conditions, like if you understand you have the right bacteria and you also understand the mechanism that you can get, then also the conditions are satisfied, the applications are there. So for example, there are a few, like if you are trying to have algae and this bacteria, it reduces the water infiltration into slopes and controls seepage. The other thing is in some other bacteria, where aerobic bacteria which are available, avoid cover for soil erosion and control and slope protection. So, the advantage is that it helps as a cover so slow protection is there. Reduce in some other bacteria like this, where you have oligotrophic bacteria in this manner, reduce drain channel erosion and control seepage. The other applications are reduce drain channel erosion, and so for example, sulfate reducing bacteria, it has grout curtains and many other things. So, if you have ammonifying bacteria, it prevents piping of dams and piping in earth dams. So, some of these applications are mentioned. So, one should be little more thorough in understanding some of these concepts. You have some more microorganisms for bioclogging. You have been mentioned here, like you can see here that you have a number of candidate bacteria for geotechnical purposes. Some of the applications include exopolysaccharide producing bacteria. This is one of the products that you get. If this material is there, exopolysaccharide, which is produced by bacteria, it is useful in reducing the permeability of the soil, selective zonal bioremediation, harbor and dam control, earthquake liquefaction mitigation. Cost of soil clogging could be reduced by using organic waste for producing exopolysaccharides, to reduce drain channel erosion, to form grout curtains to diminish migration of heavy metals and organic pollutants, to prevent. So, there are some applications mentioned. A typical application, say for example, one should understand the reactions, the enzymatic reactions, for example, using in the case of a Bacillus pasteurii to form a soil plug. A soil plug leads to reduction permeability. Naturally, soils have say for example, if I have a chemical reaction like this with you can see that it releases ammonia and then sodium bicarbonate and hydroxyls. The effect is that it increases the pH, initiation of precipitation of calcium carbonate, because in soils you have some amount of calcium, and leads to clogging of the pores and binding of the soil particles. So, the bioclogging can be used to enhance recovery of the oil from reservoirs, repair of cracks in concrete, seal unforeseen leaks in the sheet piling screens around the construction belts. And the people estimate that the cost of growing bacteria is not significant compared to other forms of, say for example, repairing of cracks with cement in concrete. It is going to be quite expensive, whereas this bioclogging could be much cheaper. So, people say that it leads to a lot of savings in costs. One should also understand that once these are all, as I said, exploratory techniques, and a lot of research is required. People have been trying to look at it, and definitely in the area of ground improvement, the sustainability has become a key issue. For example, people if there is a pile to be used, pile needs a use of steel and concrete, and then if you look at the quantity of steel and concrete that can be used in a project, and you compare with a stone column, and if both of them serve the same purpose. Say for example, load carrying capacity, if you are able to design in such a way that the load carrying capacity because of the piles is same as that due to stone columns, definitely the carbon credits that you save, or the weightage, the carbon print reduction is better in the case of stone columns, but even stone columns need aggregates, stone aggregates. So, if you have any means of reducing the stone aggregates, it is very good. And so, like that, and then if you are trying to use some say for example, instead of stone columns, you try to use waste materials in the column structure, and also try to contain them in some form, definitely it is going to be cost effective or more sensible from environmental point of view. So, there are a lot of issues, and that is why I said people have been looking at these bioremediation techniques as well for improving the ground. So, the stability of the soil after bioremediation will be achieved under favorable conditions for growth of this exopolysaccharide producing microorganisms, and there are certain things like it is not quick, but then it takes some time, and slow growth of nitrifying and Oligotropic bacteria implies long-term treatment of soil for clogging. I mean, sometimes the biomaterials need some time for reaction and favorable conditions are also there. Penetration of microbial cells in soil depth is limited by minimum soil pore size. As I just mentioned, the size of the pores is quite important, and fortunately, I think you have bacteria for every soil size. Clogging biofilm in soil pores affects the concentrations and mass transfer rates of nutrients and microbial metabolites and between the biofilm and flow through pores. So, this is an important point to be noted. Now, what is biocimentation? So, you have understood a bit of bioclogging where the permeability can be reduced. Now, what do you mean by biocimentation? Biocimentation is similar to you try to cement the particles. So, formation of soil particle binding material with the aid of microbes and additives. So, you are trying to cement the materials using this bacteria. And there is small difference. People also use fungi in bio binding, and it sometimes bio binding, what they say is using fungi is unstable and easily degradable, hence not suitable for large scale applications, whereas bacteria seems to be much better. And because in the bio cementation processes, you have the precipitation of materials in spaces between the soil particles and binds them to form a hard rock like mass. For example, X-drying of the soil particle containing iron hydroxide. Say for example, if you like to have a soil sample, increase the strength of the soil containing iron hydroxide, definitely bacteria treatment will help. Similarly, precipitation of silica dioxide, actually this is also called natural soil calcification. It also helps in cementation products. It fills pores and glues soil particles together. So, in the biocementation processes, you have a couple of activities like oxidation, reduction, dissolution, precipitation of the inorganic substances in soil, and they are strong and stable. So, the sum of the processes that we have in this thing is that what are the processes that we have in the case of cementation? Binding of soil particles with sulfides of metals produced by sulfate reducing bacteria. So, if you have sulfate reducing bacteria, so they release sulfides of the metals and then they are useful in binding. So, binding of the soil particles with carbonates of the metals produced due to hydrolysis of the urea. Then binding of particles with ferrous and ferric salts and hydroxides produced due to activity of the iron reducing bacteria. So, you have couple of materials that are required here. So, they are quite helpful in the formation of cementation. If you have calcium, magnesium, iron, manganese and aluminum, which are crystallized as carbonates, silicates, phosphates and sulfates and hydroxides, definitely there is a possibility that you get good cementation. One can get good strength in the material. For example, material plus urease producing microorganisms plus urea plus soluble calcium. It can lead to a cementing material. There is a research done on this line, like this is a urease is a material, so which can be produced by the microorganisms, and that leads to cementation. So, materials are nothing but conglomerates, breccia, sandstone, siltstone, shale, limestone, gypsum, peat, lignite, sand, clay, sediments and sawdust. If you have some of these materials and you have urease producing microorganism and all this combination, definitely it leads to high strength materials. and erase producing microorganisms are Bacillus, Sarcina, Sporolactobacillus, Clostridium and Desflotomaculum. So, of course, we must know that it is not easy to remember these names, but I think, as I said, we need to take the help of a microbiologist or a biotechnologist in this area to come out with meaningful engineering solutions. Sand enriched with culture of iron reducing bacteria, fine particles of iron ore and cellulose as electron donor demonstrate that Fe+2 form can produce cementation effects. So, in fact, they noted that the uncut compression strength can increase significantly by big number. Then how do you really use it in the field? Like, the thing is that we know that you normally compact, mechanical compaction is done, as I just mentioned in one of the earliest lectures. Mechanical compaction is normally done using shallow, it can be classified as shallow and deep, where in the case of shallow compaction use rolling or vibrating machines. In the case of deep mixing or deep compactions, you use vibro or dynamic compaction methods, and these are economical up to 10 meters deep. And dynamic compaction cannot be used for clay soil and municipal solid waste. So, when you can't do that, then the advantage is that of course, you are trying to compare them with the conventional techniques. That is what we do in mechanical compaction or mechanical modification. The other one is you can also compare them with the chemical grout, where you try to inject chemical grout into pores to enhance physical properties, and grout can be dissolved or suspended, and they reduce the permeability and increase mechanical strength. And people normally use cement, bentonite, silicates, lignosulfonates, pozzolanic based materials, thermoplastic polymers and organic polymers, etc. And we know about the method of grouting, and pressure injection grouting can be done in sandy soils and rocks. And we also have studied that the grout must have proper hardening time and low pressure is required in the case of low viscosity grout at low pressure and no change in soil volume. High pressure can, there is another classification that we already studied, jet grouting, high flow velocity mixers and grout and such, and there are a lot of issues that we already studied. But then when you are trying to use these materials, like as I said, biocementation, the biocementation requirements are as follows. Depth of penetration depends on the size of the microorganism used. For filamentous microorganisms, typical size is 1 to 3 micrometers, lengths up to 100 micrometers. It requires optimal pH, salinity, oxidation reduction potential, concentration of nutrition and water content. The advantages over the chemical grouting are that they are non-toxic. So, for example, if you look at it, lignosulfonates, acrylaminites and polyethylene or some of them are toxic chemical grouts. This actually I discussed this when I discussed about grouting, and the cost of the reagents is low. There is some cost comparison that was given in the paper that I just read, and you can see that when you are looking at chemical grouting, you have lignosulfides, sodium silicate formulations, phenoplas, acrylates, acrylamides, polyurethanes. The price is little high here, say 0.1 to 0.3, even here polyurethanes are the highest. The amount of additive required per kg, kgs in meter cube, kg per meter cube. So, it is also given, cost of additives is also given. So, it is little per dollar in terms of the dollars is given. When you compare this and look at microbial treatment, molasses plus microorganisms, the cost is somewhat less. You can say the price is 0.1 to 0.2, 5 to 20 kg per meter cube is the additives required, and the cost is 0.5 to 4, and see it is comparatively less compared to any of these numbers. And homogenized food processing waste and microorganisms, iron plus organic waste plus microorganisms, organic waste, calcium chloride plus urea plus microorganisms, you can see that they are an order shorter or cheaper compared to chemical grouting. So, where you can use them? You can use them in the enhancement of stability of the retaining walls. So, for example, if there is a cohesion that is required, normally we have a good backfill, fully draining backfill, if you want to improve the stability of the retaining wall in some sense. And of course, one should design here what is the problem actually, what are the stability calculations that one can think of. And if you are looking at retaining wall stability, embankment stability and dams, you should make a lot of calculations and make sure and if the parameters do not satisfy this stability requirements, then you need to go for improvement. And the improvement now, say for example, you are trying to improve the cohesion or the strength or the stiffness. The parameters that you need for the retaining walls and embankments and dams are the strength properties, essentially, like you want a higher strength so that the factors of safety can be higher. Then also you want deformations to be less so that stiffness can be improved. So, if you have stiffness improvement and strength improvement by using this microbial matter or the bacteria, then it is useful. And one should do a lot of experiments to confirm, yes, this improvement is there and it can be sustained for a long term, and one should understand that properly. Then the second thing is reinforcing or stabilizing soil to facilitate the stability of the tunnels or underground constructions, increasing the bearing capacity of the piled or non-piled foundations, reducing the liquefaction potential of the soil, treating pavement surface, strengthening tailings dams to prevent erosion, slope failure. In fact, in some of the places where tailing dams, they are all materials that are removed from mining, and then they have a tendency to slip away and causing a lot of risk due to slope failures, so or erosion failures. One should see that by suitable strengthening, the erosion failures are avoided. Then constructing a permeable reactive barriers in mining and environmental engineering. So, the applications are there, the binding of the dust particles on exposed surfaces to reduce dust levels, like if the bacteria can keep that dust from not going into the air, that is a good thing, and increases resistance to petroleum borehole degradation during drilling and extraction. In fact, we use certain muds to increase the borehole stability, but if any way biofilms can help in increasing the borehole stability, it is going to be nice. Increasing the resistance of offshore structures to erosion of sediment within or beneath the gravity foundations or pipelines, particularly in offshore structures. Because of the erosion, there is a possibility that the foundations of the pipelines and other structures will have a serious problem. So, if we can increase by introducing some biomaterials, there is an increase in strength or stability, then it is going to be good. Stabilizing soil pollutants from soil by the binding. Of course, the movement of the transfer of or the pollutants can be reduced by this material. Controlling erosion, erosion is something that is very important. Creating water filters and borehole filters, immobilizing bacterial cells into a cemented active biofilter. These are all some applications that one can have, and one should understand that the process of microbiology are a little slow, and it depends on complex factors like pH, concentration of donor and acceptors of electrons, temperature, concentrations of diffusion rates of the nutrients and metabolites present. Apart from the soil conditions and the grouting medium content, microbiological, ecological and geotechnical aspects must also be considered. Bioclogging and biocementation require the data of growth of the bacteria, the biosynthesis, the mechanism of biosynthesis, the biodegradation, the bioreduction, biooxidation, specific enzymatic activities, precipitation, crystallization, addition. So, these are all some important inputs, like say when you add bacteria into soil, you must be able to understand what is the growth of bacteria and how is it going to react with the soil. So, some of these various mechanisms are listed here, such as biosynthesis, biodegradation, bioreduction, biooxidation, there could be specific enzymatic activities. All these things should be understood. Even precipitation of certain salts are also possible. So, people have done at a laboratory scale some of them, but not much laboratory work, big scale work was done. A big field test was not attempted, and hopefully people take it seriously. This is just the beginning. Maybe the serious work started only about 4-5 years back. People have been working on these lines, and in Japan and in Singapore and in places like US. Assumption derived from general consideration of physiological activity of prokaryotes. So, bacteria with gram-positive type of cell wall are most suitable for soil bioclogging and biocementation. These bacteria are most resistant to changes of osmotic pressure, which is typical condition of soil on construction sites. So, definitely if the bacteria are able to satisfy these two requirements, definitely they are going to be very helpful. Then screening of microorganisms. You need to identify the group of chemotrophic prokaryotes which have wide physiological diversity. Growth only on soil surface and produces rigid crust, which diminishes soil infiltration and improves slope stability. So, lower layers become anaerobic and hence less efficient. So, one should understand that there are many processes in screening, and so for example, if you have anaerobic fermenting bacteria, they should need to be used when calcium and magnesium or ferrous ions are present. Cementation is due to increase in pH caused by ammonification and carbon dioxide production in soil added with urea or waste protein. If carbohydrates are added to the bacteria, the bacteria diminishes the pH and causes precipitation of silicates. This is another one. So, anaerobic fermenting bacteria are, they cannot clog soil pores by synthesis of extracellular polymers, because they cannot produce large quantity of slime. Organic acids, hydrogen and alcohols can be used as donors of electrons, and some of this can be used for the wastewater treatment as well, or even the groundwater. So, you have sulfase reducing bacteria, as I just mentioned earlier. They produce dihydrogen sulfide using organic acids, hydrogen or alcohols as electron donors, and sulfate as electron acceptor. Sulfide reacts with iron and other material to form insoluble sulfides of metals. These clogs the soil pores and binds the soil particle. The soil compaction created is unstable because they can be oxidized to sulfuric acid or sulfates. Most suitable bioagents are ones that produce big quantity of exopolysaccharides, which usually promote formation of the cell aggregates. They can work in both aerobic and anaerobic conditions. Both microzones of aerobic and anaerobic coexist. There are some examples here: X alkaligens, Enterobacter, and some of them given here. Aerobic bacteria, they produce big quantities of slime, form chains, filaments, and increase pH and oxidize different organic and inorganic substances. You have actinomycetes are also used. pH increases due to ammonification, and then insoluble carbonates are precipitated, forming particles that clog the soil pores. Formation of anaerobic zones below the dikes into bacteria inactive. Below the formation of anaerobic zones below the, makes the bacteria inactive. So, there is some way of screening the mechanisms here and relation to oxygen and type of energy generation. Of course, these are all taken from literature. One needs to really do some experiments on this, one can. So, very important thing that I just mentioned earlier was that the biosafety. The most suitable microorganism for biocementation, bioclogging must be safe. Like if you are trying to use it, it should be safe. It can be solved by selecting a safe bacterial strain. The biomass of this safe strain can be produced in a bioreactor and used as a starter culture for bioclogging or biocementation. And to diminish the risk of pathogenic accumulation, the following conditions are used. An application of carbon sources which are used in nature by saprophytic microorganisms such as cellulose containing agricultural waste, vegetable processing waste, etc. Conditions that are suitable for the growth of autolithotropic bacteria. Carbon dioxide is used as carbon source and inorganic substances are used as electron donors. The conditions that are suitable for application of bacteria able for anaerobic respiration with sulfate and ferric ion as electron acceptors. The application of some of these low carbons are also quite useful, and it is important that the bioclogging and biocementation processes could be used to improve the mechanical properties of the soil in situ. They can replace energy demanding, expensive and environmental unfriendly methods with significant reduction cost. And I must also say that experiments that are conducted by in the Chu and other authors, they clearly indicate that there is a permeability of the order, the reduction of the order. Say for example, instead of -4 centimeter per sec, -2 centimeter per second, it became -4, which means an order of 100. Even there was a good strength increase. Most suitable microorganisms for large scale construction and environmental problems are facultative anaerobic and microaerophilic bacteria. So, what it is clear is that these bacteria are more general and one can use them for large scale construction, and industrial scale applications of microorganisms in geotechnology is not yet started, and likelihood is that the some of these, though they are on experimental scale, if they go to commercial scale, the possibility is that they can be an industry within themselves, in the sense that similar to a ground improvement technique companies that you have for grouting, one can specialize in trying to understand what type of soil you have in a particular application and look at what type of microbes should be used, what should be are the conditions sufficient, and if the conditions are sufficient, how can you, you should do actually a field laboratory experiment and then come out with the dosage and other requirements and environmental conditions and find out the optimum benefit. Say for example, if you are reducing the permeability, you try to come out with laboratory values of reduction permeability as compared to the original value, or if you are looking at strength improvement, look at what exactly is the order of strength required, and also do a laboratory count form of the number of laboratory tests and try to replicate the same in the field trials as well before one can go for field application. So, they have large scale industrial applications, one people need to see. With this, I would like to say that the biocementation and bioclogging have a significant role to play in the development of what is called sustainable geotechnical engineering practice, and considerable research is required. Though it has been done at a limited scale in many places, there is a lot of scope for geotechnical engineers to understand bio matter and material characteristics, and that is only possible in association with a suitable group of engineers who are also trying to give some inputs into the whole system, and with the result that you have improved bio cementation or bioclogging systems that can be used for field practice. The other point that I would like to just mention here is that though there is an emphasis on biomatter here, I would like to mention that in the earlier lectures also I was mentioning about the use of natural fibers, say for example, or natural geotextiles for increasing the slope stability. And say for example, there is a steep slope of about 60 degrees, and definitely because of the rainfall there is an erosion of the slope, and definitely use of the geotextiles, natural geotextiles, say for example coir or a jute geotextile, can help in that process. And similarly, I was seeing certain applications of certain root penetration. You try to identify certain plant species, and one can say that the roots will penetrate into the soil. What happens during the erosion is that a thin layer of soil of, say for example, 32 centimeters comes out, and that once some soil starts coming out, it leads to large scale reductions, and finally there is a whole instability region sets in, and then seepage starts and all that. The first step would be in such case is to arrest the erosion itself per se, and that is only possible by erosion control mats, which are being practiced in many places. And in northeast or in some other places, people have been using specific varieties of plants. So for example, vetiver is something that people have been using in northeast on trial basis, and people claim that the penetration of the roots can be to the extent of about half a meter, so that the half, and then the rate of penetration of this could be about 3 to 4 months or something. So and then if you start in a season where the well ahead of the rainy season or something, the possibility is that the roots can grow in that time before the rains set in, and you may ensure some sort of stability. And a couple of issues are also more important here when you are trying to choose a particular plant species. Again, you need to have the density, the type of material, then the density of plantation, then their effect. One should do laboratory studies also, and also field studies before you go for large scale application. What happens most of the time is that some of the I have seen certain cases, sometimes the as I said, biomaterials or biostabilization takes some time. So it is better that even some a combination of regular ground improvement techniques and the biosystems are used. I will give an example. You have a say 40 degree or 45 degree slope, and there is lot of rainfall. Definitely 45, see the slope may be unstable, say for example for a particular in an earthquake prone area, and what you should do is that to arrest deep seated failure, say for example to the extent of 2 to 3 meters or beyond, 3-4 meters, one should use techniques like soil nailing, which can increase the deep seated stability. And if you are trying to increase the superficial stability, one can go for either the biomaterials that I just mentioned about the growth of bacteria and also the growth of plants. So some of these combinations are going to be definitely helpful, and increase in it's a combination. See, it is very difficult to completely have a sustainability ensured, in the sense that there is lot of gap already. Say for example, these techniques are not well proven. One problem is that people have been talking about the biotechnical stabilization concepts, except in the case of slope stabilization of shallow slides, it has not taken significantly. And there are also some research that is done. So for example, how to reduce the settlements, reduce the settlement where, say for example, you take soft soil, and then the soft soil has a tendency to undergo lot of settlements, but definitely that order of settlement, if you want a particular level of strength, the soil needs to undergo a lot of strength, deformation. One way is that introduce certain matter like a biomatter, and then see that the biomatter clogs the soil, occupies the soil pores or even binds the soil particles, such that when you apply the load, the stiffness is higher, and that can lead to reduction in settlement, and then you can strike a balance between applied loads. Say for example, in a particular ground improvement project, the applied preload could be of the order of, say for example, 100 kPa. If you are able to reduce surcharge at the preload to maybe 10 kPa, it is a big difference, and if you are able to identify for the 90 kPa improvement suitable biological treatment in the form of either the bacteria, it is going to be very, very beneficial. So, what I would like to say is that the area, the working on sustainability aspects in ground improvement has picked up, and people need to really do a lot of experiments and see that, do field trials as well, and see that they have come out with guidelines. For example, particularly the biomaterial, as I just mentioned, they are site specific and they need lot of conditions. What is applicable in a particular area cannot be applied in something else, because the conditions could be different. So, with this conditions, one should really conduct field trials. Apart from laboratory trials, field trials are essential for before one attempts large scale applications. So, with this I personally feel that we have come to a very broad understanding of various aspects of ground improvement in this 39 lectures that I conducted so far, right from need for ground improvement, and then we discussed about various methods such as mechanical stabilization of mechanical modification, thermal modification, then hydraulic modification, modification by inclusion such as reinforcement. Then we had extensively covered the use of geogrids, geosynthetics, soil nailing, micropiles, and also we discussed about the use of biotechnical stabilization systems and all that. So, I would like to maybe in the next class, which is the last lecture, I would like to cover summarize, give some more information on the how the further how new techniques, so the thing is that new techniques are also necessary, like maybe I will just discuss on nanotechnology and its applications. In the 3 lectures that I was just addressing, 38th lecture we discussed about the geoengineering and then trying to recover heat from the ground, which is nothing but, in fact, the ground improvement area has taken a new meaning, that you are trying to say that geoengineering. Nowadays there is a lot of discussion on how to handle the ground and how to make use of the best of the natural resources. So, in the last 3 lectures, last 2 lectures particularly, I was talking about geothermal energy and the systems that are required, and today we discussed about the use of biological or microbiology systems into geotechnical engineering or ground improvement. And I will also discuss about nanotechnology in this next lecture, and and then we will just see that I think there is a lot of scope for nanotechnology in ground improvement, and we will give some case studies as well. So, with this I conclude. Thank you very much.